chapter 15 is really going to focus on gene expression. So we'll talk about why gene expression matters. We'll talk about it in bacteria. Then we'll talk about it in eukaryotic organisms. We'll talk about epigenetics and combinatorial control and how we can actually vary gene expression. And then we'll talk about some techniques in order to actually visualize gene expression. So really, gene regulation helps cells to control their level of gene expression, and it ensures that proteins or RNA are produced at the right time and in the right amount. So multicellular organisms need to express their correct genes for a particular cell type, and they need to do that at the correct stage of development as well. Single-celled organisms need to conserve energy and, make the right protein products at the right time. So that's important for both conserving energy and cell differentiation. Natural selection really favors organisms that produce only what they need at the particular time they need it. So bacteria can regulate gene transcription. If we think about a metabolic pathway, remember that a metabolic pathway is going to require multiple enzymes, and the genes for the enzymes can actually be clustered together on the bacterial chromosome and controlled by a single promoter. So then transcription would produce one really long mRNA that codes for all the enzymes that are needed for that pathway. So in this picture below, we've got this section of the chromosome. There's a promoter, and this promoter controls structural gene A, B, C, and D. And then that produces one big long mRNA, and that one big long mRNA codes for protein A, protein B, protein C, and protein D. So bacteria have something called operons. So those clustered genes can be controlled by a single kind of on or off switch, and that switch is a segment of DNA called the operator, which is usually positioned within the promoter. So an operon is the entire stretch of DNA that includes the operator, the promoter, and the genes that they control. And there's two main types of operons: repressible and inducible. So we're going to go over an example of each of those types. A repressible operon is one that is usually on, but the binding of a repressor to the operator is going to shut off transcription, so it could be repressed or turned off. The trip operon is an example of this. So if tryptophan is absent, the repressor is inactive, so the operon is on and the cell can build tryptophan. However, if the cell already has enough tryptophan, why would it need to make more, right? So in this case, the repressor is going to be active and the operon would be turned off. So repressible enzymes are usually going to function in anabolic pathways. Remember, anabolic pathways are going to build molecules, and so their synthesis is repressed by high levels of the end product of that pathway. So here's a picture. It's a good kind of simplified picture here. So we've got our RNA polymerase binding to the promoter region because the repressor is inactive because tryptophan is not present. And so since RNA polymerase can bind to the promoter, it's going to produce mRNA that codes for the various proteins that are involved in the synthesis of tryptophan. However, when tryptophan is present, why would you need to make all the enzymes required to make tryptophan? That's a whole waste of energy. So tryptophan can bind to the repressor, which blocks the RNA polymerase binding to the promoter region. Here's the picture from our book, which is a little bit more complicated here, but they're showing the regulatory gene, and that codes for the repressor here. And when tryptophan is absent, the repressor is inactive, so the operon is on, so RNA polymerase can bind, and it produces the mRNA that codes for the proteins to make tryptophan. And then when tryptophan is present, the tryptophan can bind to the repressor, and then that repressor binds to the operator, and that blocks RNA polymerase from binding, so you don't get any mRNA and you don't get any of the enzymes required for tryptophan synthesis. So an inducible operon then, an example here is the lac operon. An inducible operon is one that's usually off, but when a molecule called an inducer inactivates the repressor, it turns on transcription. So in this case, that allows the cell to use lactose. So if lactose is absent, the repressor is active and the operon is off. Like, why would the cell make the molecules to use lactose if there's not any lactose available? That would be a waste of energy. However, if lactose is present, the repressor is going to be inactive, so the operon will be on. So inducible enzymes are usually going to function in catabolic pathways. So here's kind of a simplified picture again. So in the absence of lactose, the lac repressor is going to bind to the operator and transcription is going to be blocked. However, in the presence of lactose, the lac repressor is going to be released from the operator and transcription is going to proceed. So RNA polymerase can bind to the promoter and this is in bacteria, but the bacteria can produce the enzymes it needs to metabolize lactose. So here again is our books version. This is a little bit more complicated, but it's showing you the regulatory gene sequence here, the mRNA that codes for the protein, that is the repressor. So if lactose is not present, the repressor is in place here and RNA polymerase cannot bind, so you don't get any mRNA made and you don't get any enzymes that can deal with lactose. However, if lactose is present, the allolactose can bind to that repressor, and then the repressor is going to be removed from the operator, so RNA polymerase can bind to the promoter region and the DNA can be read to produce an mRNA, and that mRNA can be read to produce the enzymes that help the cell use lactose. So an inducible operon, again, is one that's normally off but can be turned on. So genes for bacterial enzymes in a particular metabolic pathway are often far apart or close together. So that's going to be close together. And which type of operon codes for proteins usually involved in catabolic pathways? So that's going to be inducible. And then which type of operon is usually on but can be turned off? So that's going to be repressible. So eukaryotic organisms then can also control their genes. We say that eukaryotic gene control is combinatorial, meaning a combination of many factors regulate gene expression. So kind of an everyday example of that is like volume control on like your computer. So if you have like a desktop computer and you're listening to music on Spotify, you've got the volume control, on Spotify or Pandora if you'd rather, right? And then you've got the volume control on the computer itself and then on the speaker. So you've got 3 ways to control your volume settings. So broadly speaking, positive control is going to increase transcription rate, and if we increase transcription rate, we have more mRNA, so we'd have more proteins, and the negative control decreases transcription rate, so we would have less mRNA, and so we'd have less proteins. So there's several targets for gene regulation, and your book really does a nice job laying this out. So I took this picture from your textbook. I highlighted in purple the ones that we're going to actually kind of focus on or worry about. So chromatin modification, transcription, RNA processing, transport to the cytoplasm, translation, protein processing, degradation of the protein, and transport to the cellular destination are all things that can be modified. So as far as regulation of chromatin structure goes, you know that eukaryotic DNA is packaged into chromatin, and we talked before about nucleosomes where DNA is wound around these histone proteins. However, DNA can be wound tightly or loosely around those histones. If you went to that Learn Genetics site and licked the rat to go over epigenetics, you would be able to see an example of that. Histone acetylation is one thing that can make the DNA less tightly wound around histones, so that would be an example of positive control. And then DNA methylation is when methyl groups are attached to DNA at this base cytosine, and that's going to close chromatin structure, so that would be an example of negative control. As far as epigenetic inheritance goes, remember this is the above the genome inheritance, so this chromatin modification can actually be passed on to future generations of cells. So the nucleotide sequence itself is not changed, however, the packaging of the DNA is changed, and so that can be passed on to offspring. I've found you guys a diagram from a journal article that kind of gets a little bit more in depth in this than you really need to know, but if there's some sort of environmental insult or life experiences or social interactions that modify the chromatin conformation, that can actually be passed on to the organism's offspring. However, it can also be reversed, so it just really kind of depends, and this is a really exciting area of research that we're finding more and more about. So if we talk about regulation of transcription initiation, we can target RNA polymerase. Remember, RNA polymerase is actually going to make the piece of mRNA, so activators can bind to enhancer DNA sequences and make it easier for RNA polymerase to bind, so that would produce more mRNA, so that would be positive control. Repressors are proteins that can bind to silencer DNA sequences, which makes it harder for RNA polymerase to bind. So that would be an example of negative control, and there would be less mRNA produced there.
Most of the fine-tuning of gene expression occurs at the transcription level, making an RNA copy of a DNA sequence. Recall that to initiate transcription, an RNA polymerase must bind to the promoter region adjacent to a gene. This cannot occur without the help of specific protein transcription factors. Some transcription factors, called activators, bind to enhancer regions some distance from the gene. The DNA bends, bringing the activators closer to the gene, where the activators interact with other transcription factors and RNA polymerase to initiate transcription.
So then it's also possible to do post-transcriptional regulation. So remember, transcription is when we go from DNA to RNA. So in this case, we can do RNA processing, again, the 5' cap and the poly A tail, or we could do alternative splicing, which we talked about in the previous chapter. We can also alter translation initiation, so proteins may bind to the untranslated regions like the 5' cap or the poly A tail and either activate or prevent translation. And we can also adjust the lifespan of the mRNA in the cytosol. So if the mRNA is around longer, more proteins can be produced. If it's around a shorter period of time, less proteins could be produced. So often non-coding mRNA can interfere with mRNA. So if we look at those non-coding mRNAs, microRNA can bind to complementary mRNA sequences, and this is either going to degrade the mRNA or block translation. There's also small interfering RNAs that are actually derived from two RNA molecules that come together to form this kind of double-stranded region, right? We told you RNA was single-stranded, but it can be double-stranded, and that can also block translation. And it's actually involved in chromatin packing in yeast, which is kind of exciting. But both microRNAs and small interfering RNAs are going to block gene expression by RNA interference, which is a great name for this, right? Because it's interfering with the RNA, so it's called RNA interference. As far as post-translational processing, remember translation is when we've actually created that protein. So proteins can be processed by folding, which I think is pretty straightforward as far as like the idea of what folding is, or cleavage, where the proteins can be cut into pieces, or phosphorylation, where we add a phosphate group, or the addition of sugars as well. Or proteins can be tagged with ubiquitin to trigger their destruction and breakdown. So I found you guys a few pictures of this because our book was pretty lacking in those pictures. I actually got those from journal articles, so if you're super interested, you can go to those links there. This next table, we're going to go over this in class together, but I would encourage you to kind of take a look at it and see where you are stuck on that, or if you're stuck, and feel free to come and ask me questions. So then how can researchers actually observe gene expression? Basically what they're going to do is look for the mRNAs in the cells, because if that cell has the mRNA, it likely has produced that protein. They can also look for the protein product, but we can look at the mRNAs and tell as well. So there's a couple different techniques they can use. They can use nucleic acid hybridization, where they use fluorescently tagged nucleic acids to kind of probe and bind to complementary sequences, and then we can see what's fluorescing in these organisms. They can also use reverse transcriptase polymerase chain reaction, which is totally a mouthful to say, but it's called RT-PCR. We previously talked about polymerase chain reaction, or PCR, which was used to make a whole bunch of copies of DNA from DNA. In this case, with reverse transcriptase polymerase chain reaction, or RT-PCR, if we think about what transcription is going from DNA to RNA. So if we did that in reverse, it would be going from RNA to DNA. So this is going from RNA to DNA and then making a whole bunch of copies of DNA. So this turns mRNAs into double-stranded DNA molecules with the enzyme that's called reverse transcriptase, and that produces cDNA. The C in that stands for complementary DNA, and it's cDNA because it's complementary to mRNA. DNA microarray is another way we can look at gene expression. In this case, you're going to have multiple known DNA fragments that are fixed to a single slide. Sometimes this is called a DNA chip or a gene chip. So then you're going to isolate mRNAs from a cell of interest and create fluorescently marked cDNA, and then you're going to allow the cDNA to hybridize with the DNA on the chip. And you can compare gene expression in different cell types by marking them with different fluorescent. So let's match the images here. The first one, we're comparing cancer cells to normal cells, and we're looking at gene expression using red or green fluorescent probes. So the first one is going to be microarray, and they've got the little gene chip there in the picture as well. The next one, we're starting with RNA, and then we're making a strand of cDNA, and then we're making a cDNA hybrid, and then we're making a whole bunch of copies with just regular old PCR. So that's going to be RT-PCR. So chapter 15 was relatively short and sweet. We talked about differential gene expression and how bacteria and eukaryotic organisms control gene expression. Make sure you know the difference between reducible and inducible operons. Get a big picture idea on epigenetics and the different targets for regulation of gene expression and the techniques we can utilize to measure gene expression in cells.